What if I told you that the key to keeping livestock alive in like one of the most extreme oxygen-starved environments on Earth is actually the exact same plant you might use to cure a Sunday morning headache?
I mean, it sounds completely counterintuitive, right? You have this incredibly harsh, unforgiving landscape, and the solution isn't some, synthesized chemical from a futuristic lab.
Yeah.
It's literally a leaf that indigenous populations have been boiling in water for centuries.
It's wild. Well, welcome to another customized deep dive. Today we are taking you to the very edge of where agriculture is even physically possible.
Exactly.
We're looking at this fascinating intersection of extreme high-altitude survival, ancient botanical medicine, and modern farming.
And our roadmap for this conversation is a pretty groundbreaking research article. It's titled Effect of Salix alba Leaf Extract on Growth Performance, Antioxidant Level, and Immune Status of Broiler Chickens Reared at High Altitude Cold Desert.
Quite the mouthful. But the mission for our deep dive today is to explore how researchers are taking this ancient medicinal plant and applying rigorous modern science
Yeah.
to solve a massive problem in Livestock.
Right. And for those of you listening who might not be familiar, Leh-Ladakh is a region in the Himalayas sitting at a staggering 3,500 meters above sea level.
Which is roughly 11,500 feet.
Yeah, to put that altitude in perspective for you, it is an absolute biological battleground. I mean, the very air you breathe changes.
Oh, for sure.
So the first major hurdle for anyone trying to raise poultry there is hypobaric hypoxia.
Hypobaric hypoxia. So we are talking about two things combining here, right? Low atmospheric pressure and low oxygen.
Precisely. It isn't just that there is less oxygen in the air,
Yeah.
The atmospheric pressure is so low that it physically makes it harder for the lungs to drive what little oxygen there is into the bloodstream.
Wow.
Every single breath yields just a fraction of the oxygen you would get at sea level. But that is only the first stressor.
Right, because farmers in Leh-Ladakh are also dealing with temperatures that can fluctuate violently. I mean, from a baking positive 35 degrees Celsius in the summer to a bone-chilling minus 35 degrees in the winter.
Yeah, the extremes are insane.
And then you add in intense ultraviolet radiation because the atmosphere is thinner, incredibly low humidity, which, just dries out the respiratory tract, and then nutritional stress.
Because it's so hard to transport high-quality feed up the mountains, yeah.
Exactly. I guess my first real question is, why even try? Why are they trying to raise broiler chickens in an environment that actively seems to want them dead?
Well, it really comes down to basic food security. You have local populations and often large military presences in these border regions that require a reliable source of protein.
Oh, that makes sense.
Right, and transporting live birds or frozen meat up mountainous terrain is incredibly expensive and logistically fragile, so local production is vital.
But for a broiler chicken, which has been bred for a massive metabolic rate and rapid growth, this environment must just drastically reduce their health and survivability.
Oh, absolutely. It reduces their productivity massively, which means devastating economic losses for the farmers trying to provide that food.
Right. But to really understand why these birds struggle so much,
Yeah.
we have to look past the weather and look at what this environment does on a microscopic level, right?
Yes. We have to look at the ultimate physiological villain in this environment, which is oxidative stress.
Okay.
This is the actual mechanism that breaks the birds down from the inside.
I always think of oxidative stress as biological rusting. Like if you leave a piece of untreated iron out in the rain, it reacts with the environment, it rusts, and it loses its structural integrity.
I actually really like that analogy because chemically it's very accurate.
Oh, really?
Yeah, under normal conditions, any living body produces a small amount of reactive oxygen species, which are often called free radicals. They're just a natural byproduct of cellular metabolism.
Okay, so just normal bodily functions.
Right. Now, imagine a free radical as a molecule that is missing an electron, which makes it highly unstable.
So it wants to find an electron to stabilize itself.
Exactly. And it will violently steal that electron from wherever it can, from the lipids that make up cell membranes, from essential proteins, even directly from the animal's DNA.
Yikes. So that electron theft is the rusting we were talking about.
Yes. And normally, a body's antioxidant defense system easily neutralizes these free radicals by willingly handing over an electron without becoming unstable themselves.
But at 3,500 meters, with the low oxygen, the freezing cold, and the high UV radiation, I imagine that normal defense system just gets completely overwhelmed.
It gets entirely swamped. I mean, the environment forces the bird's metabolism to work in overdrive just to maintain body heat, which creates a flood of free radicals.
Oh, wow.
Yeah, and when this biological rusting happens on a systemic scale, the immune system becomes incredibly suppressed.
So the birds literally can't utilize their feed efficiently because their cellular machinery is too busy trying to repair the constant damage.
Exactly. Growth stalls and mortality rates just skyrocket.
So improving their internal antioxidant defense system isn't just like a nice little bonus. If you want these birds to survive, you have to provide them with external antioxidant reinforcements.
Right. And for decades, the agricultural industry's answer to keeping livestock healthy under stress was to lean heavily on synthetic feed additives and antibiotic growth promoters.
And this is where I want to challenge the premise of this study a little bit.
Sure.
If a farmer is facing, say, 50% mortality in their flock, why wouldn't they just use modern synthetic medicines or antibiotics? Like, why are researchers looking backward at ancient plants instead of forward at new pharmaceuticals?
That is the exact question the global agricultural sector is wrestling with right now. The problem is that the old modern way is failing us.
How?
Well, the routine use of antibiotics in livestock feed is a massive driver of antibiotic resistance. We are creating superbugs that actually threaten human health care.
Oh, right.
Plus, there is growing consumer demand to eliminate synthetic chemical residues in meat. Because of this, science is pivoting hard toward natural, what we call phytogenic alternatives.
Okay, so phytogenic meaning derived from plants. Which brings us to the star of this research, Salix alba, commonly known as the white willow.
White willow is fascinating. It's been used in traditional medicine systems across Europe and Asia for thousands of years, specifically to treat pain, inflammation, and fever.
It's basically the ancient precursor to modern aspirin, right?
Yes, exactly.
The leaves of this tree are absolutely packed with bioactive compounds. Like the researchers were looking at things like salicin, caffeic acid, polyphenols, flavonoids, and tannins.
And the reason those specific compounds are so valuable is that they possess powerful antioxidant and anti-inflammatory properties.
So the researchers basically wanted to know, if we take this ancient fever medicine and adapt it for modern agriculture, can it serve as a phytogenic shield against the extreme stress of the Himalayas?
That was the mission. They wanted to evaluate if a highly concentrated water-based extract of these white willow leaves could actually improve the growth, immunity, and economics of raising broilers under hypobaric hypoxic conditions.
But you don't just grab a handful of leaves, throw them in a bucket of chicken feed, and hope for the best, right?
Definitely not. No, the rigor of the preparation is what separates anecdotal folk medicine from reproducible science.
I was actually marveling at the extraction process while reading this study. They sent researchers directly into Ladakh to collect fresh leaves just to ensure the plants were native to the environment.
Which is a crucial first step.
But then they shade-dried the leaves for 4 to 5 days. Why shade-dry them? If you're in a cold desert, wouldn't you want to put them in the direct sun to dry them faster?
That's a really great question. You actually want to avoid direct sunlight specifically because of the intense UV radiation at that altitude.
Oh, because it would damage the plant.
Exactly. UV light can degrade and break down the delicate phytochemicals inside the leaf. By shade drying them at room temperature, they slowly remove the moisture while preserving the medicinal integrity of the plant.
That makes perfect sense.
Yeah.
So they dry them, grind them into a powder, and then they use something called a sock slit apparatus.
Ah, the sock slit. It's a brilliant piece of classic chemistry equipment.
What does it do?
Well, imagine a highly precise, continuous espresso machine. They use distilled water at about 80 degrees Celsius, continuously washing it through the powdered leaves for 24 to 48 hours.
Oh, wow.
This method ensures they pull out the absolute maximum amount of those valuable water-soluble phytochemicals without burning them.
Okay, so then they filter it, remove the excess water, and finally, they freeze-dry the extract at nearly minus 90 degrees Celsius. I'm assuming the freeze-drying is for the same reason as the shade-drying, avoiding heat damage.
Exactly right. Freeze-drying locks the compounds in a stable, powdered state without applying destructive heat. The final result was a perfectly standardized extract yielding roughly 36% dry matter.
And that standardization is crucial. Right?
Vital. If you're going to use a plant as a medicinal additive, you have to know its exact strength so you can replicate the results on a commercial farm later.
So they have this potent brown powder, but before they even let a chicken look at it, they had to prove its biochemical worth in the lab.
Yes.
The paper, lists a whole bunch of assays, things like DPPH and ABTS. For you listening, if your eyes just glazed over at the alphabet soup, essentially these are tests to see how strong the extract's antioxidant shield really is.
Yeah, that's the perfect way to look at it. Rather than just guessing that the extract has antioxidants, these lab tests are like a simulated battlefield.
How so?
The researchers introduce a known quantity of synthetic free radicals into a test tube with the extract, and they measure exactly how many of those free radicals the extract can neutralize.
And across the board, the White Willow extract showed massive dose-dependent free radical scavenging power. Like it proved it could stop that biological rusting in a test tube.
Right, but they also wanted to know exactly which molecules were doing the heavy lifting.
Which is why they ran an HPLC analysis, high-performance liquid chromatography. You can think of HPLC as like a molecular barcode scanner. It separates the extract and identifies exactly what is inside and in what quantities.
And the span confirmed that a compound called salicin was the undisputed star of the show.
Yeah, the HPLC clocked it at over 790 micrograms per milligram of extract. They also found caffeic acid, but salicin was the heavy hitter.
Salicin is incredibly important biologically. In the body, it acts as a very potent natural anti-inflammatory. It works primarily by inhibiting the synthesis of prostaglandins.
Okay, wait, let's pause there for a second. Prostaglandins, how do they fit into the picture?
Think of prostaglandins as the body's chemical alarm bells. When cells are damaged, say by extreme cold or low oxygen, the body synthesizes prostaglandins to trigger inflammation, swelling, and pain to protect the area.
Oh, so it's a useful defense mechanism in short bursts.
Exactly. But under the constant stress of the Himalayas, those alarm bells never stop ringing. The chronic inflammation completely exhausts the bird.
Oh, I see.
By inhibiting those prostaglandins, salicin essentially turns off the blaring alarm, reducing the systemic stress on the body.
So the extract has proven its chemical potential, but we're still not at the farm yet. The researchers took this to the Petri dish next, which I found totally fascinating.
Yeah, the in vitro phase.
Right, they isolated peripheral blood lymphocytes directly from chickens. Why stop at this step? If you know the chemistry works, why not just feed it to the birds?
It's a combination of ethical science and mechanism verification. Lymphocytes are key white blood cells, the frontline soldiers of the immune system. Before doing a massive feeding trial, you really want to see exactly how the extract interacts with living immune cells in a completely controlled environment.
And the results were a twofold victory. First, just exposing the cells to the extract caused them to multiply and thrive. It was like fertilizer for the immune system.
Yes, it actively promoted lymphocyte survival and proliferation. But the second part of the Petri dish test was even more relevant to our high-altitude scenario.
Right, because they intentionally induced oxidative stress on these cells using hydrogen peroxide, basically mimicking the harsh environment of Livestock right there in a Petri dish.
And the extract actively protected the cells from the hydrogen peroxide.
It's like the extract acts as a highly effective bodyguard, taking the hit from the oxidative damage, while simultaneously acting as a personal trainer, strengthening the immune cells before the real fight begins.
I love that. It's a great way to visualize it. Having established that strong immunoprotective potential in living cells, they were finally ready for the ultimate test, the in vivo trial.
The 42-day high-altitude field experiment. The setup was super ambitious. They took 105 one-day-old broiler chicks and divided them into 7 groups, all raised in these controlled, extreme high-altitude conditions.
Right, one group was the control, meaning they just got standard high-altitude feed with no extract at all.
And the other 6 groups got the same diet, but supplemented with different doses of the White Willow extract, ranging from 100 milligrams to 800 milligrams per kilogram of body weight.
And they meticulously monitored everything for those 42 days. Body weight, feed intake, the feed conversion ratio, blood biomarkers, mortality rates, and of course, the financial bottom line for the farmers.
Now, if you're listening to this and you've ever taken daily vitamin C, you know the temptation. If you feel a cold coming on, you think, I'll just take a massive handful of vitamin C pills. More is obviously better.
All right, human nature.
Yeah, so you'd assume that the group getting 800 milligrams of the extract would become invincible super chickens.
It is a very common assumption, but biological systems rely heavily on balance. The study revealed a very clear Goldilocks dose.
So more wasn't better.
No, there was a sweet spot where the physiological benefits peaked without overwhelming the system. That dose was 300 milligrams per kilogram of body weight. The Salix 300 group.
Okay, let's talk about the results for that Salix 300 group because the numbers are just staggering. Starting with growth. The feed conversion ratio, or FCR, is arguably the most important metric for any farmer.
Oh, absolutely.
It measures how much feed it takes to grow 1 unit of animal weight. In the control group, the FCR was 4.73.
Which is exceptionally poor. It basically means the birds are eating massive amounts of expensive feed, but they aren't actually gaining weight. It reflects how much energy they are wasting just trying to keep their organs functioning in the cold and hypoxia.
But in the Salix 300 group, the FCR dropped to 3.48. That is a huge improvement. The birds grew significantly heavier, using the exact same amount of feed and water.
Yeah, their bodies just use the fuel vastly more efficiently.
To understand how they unlocked that efficiency, we have to look back inside the body at those internal mechanisms. Remember our analogy of the biological rusting, the free radicals tearing apart the cell membranes.
Yes, the lipid peroxidation.
Right.
So the researchers measured a biomarker in the blood called MDA. Think of MDA as the chemical footprint left behind when free radicals successfully destroy lipids.
Okay, so it's a measure of the damage.
Exactly. In the control group, MDA levels were extremely high. The rusting was severe. But in the Salix 300 group, MDA levels plummeted. The antioxidant shield provided by the White Willow was actively holding the line.
Wow. And the physiological benefits didn't stop there either. The coolest internal shift was what happened to their immune systems. There was a complete rewiring of their cytokines.
Oh, definitely.
And for you listening, cytokines are basically the messenger proteins that tell the immune system how to behave.
The specific shift we observed here is so fascinating. Pro-inflammatory cytokines, specifically ones called IL-1 and IL-6, significantly decreased in the treated birds.
And why is that?
This is almost certainly due to the salicin in the extract inhibiting what's known as the NFKB pathway.
Okay, we're getting into deep biology again. What exactly is the NFKB pathway?
So think of the NFKB pathway as the master fuse box for inflammation inside a cell. When the environment stresses the cell, that fuse box gets flipped on, and it commands the cell to produce those inflammatory cytokines.
Oh, I get it.
Yeah, and the salicin essentially steps in and blocks the fuse box from being flipped.
So the systemic exhausting inflammation was turned off. But they didn't just suppress the immune system entirely, did they?
No, not at all. While the inflammatory cytokines went down, another cytokine called IL-2 actually increased.
And that detail is crucial. IL-2 acts like a drill sergeant for T cells, which are the specialized soldiers of the immune system. It promotes their growth and differentiation.
Exactly. So the extract lowered the wasteful, damaging inflammation while simultaneously sharpening the targeted, effective immune response.
That is just brilliant. All of this microscopic chemistry, the lower MDA, the blocked fuse box, the sharper T cells, it all translated into very real-world results when it came to mortality.
It really did.
Raising birds at 3,500 meters is usually a tragic numbers game. The control group suffered a 53.33% mortality rate. I mean, over half the birds died.
And they were dying largely from classic high-altitude ailments. Things like ascites, which is a condition where the right ventricle of the heart fails because it's pumping so hard to find oxygen, causing fluid to build up in the abdomen.
Ugh, that's awful.
Yeah, they were also dying from intestinal diseases like coccidiosis that strike when the immune system crashes.
But in the Salix 300 group, mortality dropped all the way down to 20%. And virtually none of that was from ascites. The cardiovascular and immune systems just held up under the pressure.
And when you look at the economics, it makes perfect thermodynamic sense. Even accounting for the extra cost of extracting and adding the White Willow, the farmers saw a significantly higher net profit.
Because more birds lived.
More birds lived, and they grew better. By reducing the massive internal cost of inflammation and oxidative stress, the birds saved energy. Because they weren't burning all their calories just trying to repair cellular damage, that energy was redirected into growth and survival.
It's an incredible realization. This entire deep dive proves that ancient botanical wisdom, the traditional use of the white willow tree, when validated and applied with rigorous modern scientific methods, can actually solve deeply complex agricultural challenges.
Even in some of the most extreme environments on the planet.
Exactly. We don't necessarily need to invent a brand new synthetic chemical. Sometimes we just need to look closer at the natural toolkits that already exist.
It's a profound testament to the power of phytogenic compounds. I really think it completely reshapes how we might approach global food security in marginalized, high-stress climates as the world changes.
Totally. And it leaves me with one final kind of wild thought for you to mull over as we wrap up. We spent all this time talking about chickens at 3,500 meters.
Yeah.
But oxidative stress from extreme environments isn't just an agricultural problem. If the concentrated phytochemicals in Salix alba can protect fragile avian biology from hypobaric hypoxia, extreme cold, and intense radiation, what might these specific standardized botanical extracts mean for human applications?
Oh, wow. That's a big question.
Right. Could the exact same mechanisms that saved these birds, the salicin blocking the inflammation fuse box, the massive free radical scavenging, could that be the key to protecting human astronauts facing oxidative stress and cosmic radiation in deep space exploration?
That is a very compelling thread to pull on. I mean, human biology reacts to hypoxia and radiation in very similar ways. Nature usually scales its best solutions, whether it's on a mountaintop or in orbit.
It seems like Livestock might just be a tested ground for something much, much bigger. Thank you for joining us for this deep dive. Keep asking questions, keep exploring, and we'll see you